All right, good afternoon / good evening, everyone, and welcome to Genomics Lite. This is our Evolution and Proteins in Focus session with Zachary Arden, who's our guest speaker this evening. So my name's Em, I'm the Science Engagement Manager for Welcome Connecting Science. I'm sort of hosting and looking after the technical side of things this evening, and we've got Zachary here as our guest speaker. Before we kick off, I'll start with my sort of scene setting slide that I know a lot of you will have seen by now. So all the events that we host aims to help people explore what genomics means for their everyday lives, and obviously in this context for sort of education and careers as well. So whilst we invite you to ask questions this evening, both about Zachary's topic and about his career, we ask you to do so respectfully and sensitively as well. I'm on the call and my colleague Fran will be here later to moderate the space to make sure it's safe and inclusive for everyone. In case you somehow avoided using Zoom over the last couple of years, if you want to get in touch with us this evening, you obviously can't use your cameras or microphones because we're on webinar, but you can chat in the chat box. You can message me directly if you're having any difficulties with anything this evening. If you've got questions for Zachary, stick those in the Q&A box and we'll answer some of those as we go and at the end of the session as well. And we do have closed captions available this evening as well. So if you click the CC box, you should get a little black box with your subtitles coming up. You can also move those around the screen so they shouldn't ever really be in the way of anything. You can also turn them off if you don't want them, but they're there if you do. We're based, theoretically at least, at the Wellcome Genome campus just south of Cambridge. We're a world-leading genomics research institute combined of the Wellcome Sanger Institute and EMBL EBI. So Wellcome Sanger combines a mixture of traditional wet lab techniques as well as data techniques, whereas EMBL EBI, this B here stands for bioinformatics. They are solely computer-based, so they work completely with sort of data and databases produced by experiments. Zachary's based at the Wellcome Sanger Institute and will obviously tell you lots more about the exciting research that he does. Before I hand over, if you're looking to read more widely on sort of proteins and things relating to proteins, we've had these 4 new articles go live on our Your Genome resource website literally yesterday. So these explore what RNA is, how RNA is spliced to produce different forms of proteins, how we can sequence RNA to understand it better, and then more broadly, how do we use bioinformatics to pull lots of data together? So these are all available on yourgenome.org, which is all freely available and really useful, especially if you're revising for sort of A-levels or anything around that stage. So this evening, we'll have about a half-hour talk from Zachary about the topic of proteins and evolution. We'll then have time for a Q&A, although if there's questions coming in throughout, I'll chip in and ask those as we go. Zachary's then going to share a little on his career path, and you'll have a chance to ask questions around what it's like to be a scientist and what it's like to sort of take a science career. And we'll aim to wrap up by about 5:45 this evening. If you have to dash off early, don't worry, this is being recorded and will be available on YouTube and our website, and you'll get the link sent out to that either tomorrow or Monday. So don't worry if you have to leave early. And with that, that is my part out of the way. So Zachary, I will hand over to you.
Thanks, Em, and hello, everyone. I'll just share my screen and then we'll get started. Okay, great. So it's great to see so many of you here, and hopefully this will provoke some interesting thoughts for you. So the focus is how does evolution make new proteins? I'm a postdoc at the Wellcome Sanger Institute, and I'll tell you more about this as we go on. First, we might just want to set the scene. Why do these different things, genomes, genes, and proteins, why do they matter in the first place? Why are we even interested in this side of biology in particular? And I think this is one of the most interesting and exciting sides of biology, and I'll try and explain why. We can see these wonderful, diverse pictures of different areas of biology. And then we can think, okay, what's happening at the molecular level? So genomes is a full set of genetic information. It's made up of genes, and these genes are the instruction manuals for building proteins. And proteins are the main part of the molecular basis of life. So if we really want to understand each of these different things and many others, plants and bacteria and viruses in detail, then we'll want to understand their proteins and how they're encoded by genes and how all these genes are put together into genomes. So proteins are key for understanding the function, the functions that are occurring within the cell, diseases, what happens when things go wrong, and evolution, what's the history of these organisms, how do they come to be like they are today. So there's a whole lot that we can learn about biology if we learn about genomes, genes, and proteins. These are just the different things that I'll be going through today. So firstly, what are genomes, genes, and proteins? Just as a recap, what is evolution? In particular, what's it mean to have a molecular perspective on evolution? Some examples of protein coding genes, and then what people have typically thought about how proteins evolve. This is kind of, all of this is kind of the background to the things that then we'll be focusing on. The second part, some recent discoveries, which have changed our picture of protein evolution. I'll be focusing on this idea of how can we get a new gene and some examples from SARS-CoV-2. Okay, so firstly, what do I mean when I talk about genes? So really simply, a gene is the DNA or RNA instructions that go to make up a protein. There are different kinds of genes. We won't really be talking about those so much today. Mostly I'll be focusing on protein coding genes. And we see an example of the start of a gene here. Typically starts with an ATG codon and then additional codons, which will code different amino acids. So the protein is what's actually encoded by this gene. The string of amino acids encoded by the gene. It can fold up into a shape such as this one, which we'll come back to later. The genome is the full collection of all the genetic material in a cell or a virus. So for instance, the full genome of SARS-CoV-2 can be illustrated like this. It's about 30,000 single letters, and it encodes these different genes, which are illustrated here, which we'll come back to a little bit later. So it encodes around 15 or so genes, depending on how you count it. Okay, so just as we get started, we asked what are genes and genomes, and then the next stage is what is evolution? So this is an opportunity for you to write something in the chat and give some feedback. What do you think when you think of evolution? So I'll give you like a minute maybe to tell us what you think about that. There's already a question, but yeah, feel free to answer this one first. Feel
free to pop stuff in the chat box and I'll read some out. So we've got evolution, I think change over time. Someone's put when an organism specifically evolves. Someone's put natural selection. Someone's put survival of the fittest, and mutations is coming up a lot. So we've got lots of change over time, natural selection, mutations, Darwin, adaptation. Lots of excellent buzzwords. I like the description of some random changes that get passed down over the generations, which is a nice way of putting it. Small changes that can lead to big changes. So yeah, lots of buzzwords around that as well.
Okay, great. So I guess one thing I noticed in a lot of those answers is most of the answers that people gave are fairly small scale. But so evolution you can look at it on the small scale, but you can also look at the really large scale, the really long history of life. So one way in which I think about evolution in the big picture sense is it's the processes that were involved in going from something like this, a single-celled organism like a bacterium, the last common ancestor from which all other modern cells have arisen was around something like 3 billion years ago. It went through various processes, down different lineages in the evolutionary tree, and it ended up in different things. For instance, this turtle or this octopus, and many other things as well. So evolution, the big picture, is all the different processes that happen over billions of years. And we'll come next onto evolution, the small scale. But a question about that, which also you can answer in the chat: why might we want to use bacteria and viruses to study evolution?
Got a couple in pretty quickly. So someone's put fast replication and they evolve quickly, which I'm guessing is what we're going to get a lot of answers of. Yet they're quick. Fast reproductive cycle. A nice idea that they also might mutate more easily, and a nice sort of practical element of the easier to culture. some really good ideas about less ethical problems of studying them than studying perhaps different ones. We've got a lot coming in. I'm trying to scroll really quickly. The smaller organism, I guess that's physically and also in their genome size. They can be genetically modified easier. They've been around for longer. And someone's put about competition against each other, so bacterial phages. So loads of really, really good ideas coming in.
Okay, so we had evolution at the large scale, which is over the course of billions of years, but we can also study evolution at a much smaller scale. We can actually see it occurring before our eyes, and this is this is my favorite example, really, which is something called the mega plate experiment. I believe mega stands for microbial evolutionary growth arena. But it also is helpful because this is a really big plate. So it's about the size of like a dining room table. This is basically a massive Petri dish, a couple of meters long. And what's happening here is people have, the scientists have put bacteria at both ends of this plate, and the bacteria show up as white. And the dark region is regions where initially the bacteria can't grow, there's no bacteria there. There's just empty gel, which the bacteria can swim through, but they can't survive initially because there's antibiotic chemical in this middle region. And if you look carefully, you see this kind of columns down the plate, and each column represents a higher and higher concentration of antibiotic going towards the middle. So the bacteria initially can't grow, but over the course of a couple of days, and this is a sped up video of what's happening. Over the course of a couple of days, the bacteria can increasingly grow at higher and higher concentration. And that's because there are mutations happening in their genome, and they're able to, a small subset of the bacteria are able to survive. And then eventually another mutation occurs, and then their descendants are able to survive in the higher and higher concentration. Because these bacteria replicate once every something like 20 minutes, they can go through a lot of cycles. There's a large population size. So the population overall can adapt really quickly to the higher and higher concentrations. So this is evolution happening before our eyes, which is really cool. But what I like even more is that we can actually see what's happening at the molecular level. So we can think about the DNA, and we can actually trace the history of what's happened over the course of the experiment. So we can pull out a region of this gel, and we can find what bacteria are there. We can sequence the genomes and see actually exactly what genetic mutations happened over the course of the experiment, just a couple of days, and see which mutations were involved in allowing the bacteria to survive at higher and higher concentrations of antibiotic. So this is not a single bacteria that's doing this, but this is generations of bacteria over time. And over a few generations, you can see quite a lot of change over the whole population. So that's very quickly a nice example of the molecular perspective on evolution. What's happening at the genome level, which nucleotides are changing.
Really nice question. We just got linked to that slide that I'll ask now. So someone's asked, at what point do you classify a bacterium as a different species? So I guess on your video, like how far would it have to change from the outside ones before we say that middle one is a whole different bacterium?
Yeah, that's a really good question, and there's not really a good answer to that, unfortunately. So this is something that scientists debate a lot, and it's debated, is it kind of arbitrary what we're calling a species? At the moment, there's a certain cutoff that people use. I think I'll probably get this wrong off the top of my head, but I think it might be 97% similarity in a particular gene, the ribosomal RNA gene. And something like that is the threshold that's used in some standard approaches. And once there's enough changes occurred between organisms, and we say that those are in different bacterial species. With some other organisms, it's maybe a bit easier to define what's a species because you have like interbreeding populations, which is kind of the classic definition. Yeah, but with bacteria, it is a bit more difficult, and it is still kind of debated. So
someone just popped in the comment saying that it was is it not to do with when they can no longer interbreed? But obviously bacteria don't interbreed, so you've got to just pick a number at some point, I guess, and then just sort of decide. But yeah, thank you for that.
Yeah, that's an interesting thing because bacteria are definitely different. So like a salmonella bacteria is definitely different from an E. coli bacteria, but they're also quite similar in some ways. So where you draw that threshold for a species or a genus or a family is something that can be debated. Okay, so back to proteins, a couple of examples which you might have heard of in the news recently. So this is the spike protein, which is causing us so much problem with COVID in the virus SARS-CoV-2. The spike are these little blobs around the edge, around the outside of the coronavirus. And so we know the protein structure from lots of studies that have been done. That's one example. In viruses, here's some examples in bacteria, which I find really impressive. This is called the Bacterial Flagellum. Some of you may or may not have come across. This is basically like an outboard motor that a lot of different bacteria use to swim. So this is the long tail, and this is the motor, which rotates the tail extremely fast, above 10,000 revolutions per minute. And this is the core of the motor. And relatively recently, in the last couple of years, we've got really precise images of how each of these little units is actually a single protein. So there's something like, I don't know, 50 or 100 proteins in this ring, and a few different rings and a few different components to make this motor. We can also visualize this and actually see what it roughly looks like when it's spinning. So then this is the core of the motor, and the very long tail of the flagellum would be attached to here. So this is another nice example of what we're talking about when you think about proteins and protein molecules. So in this case, they come together to form this complex with a few different protein subunits that together form basically a machine inside the cell. Okay, how could these things evolve? How do proteins normally evolve? Well, in the very simplest case, we have something like this. This is just this block represents a gene, and we imagine that this is the ancestor of some genes which go on to exist. So we have an original gene, and then it turns out that this gene gets copied just through a chance event. This happens a lot, quite a high frequency in bacteria or yeast or in lots of other organisms. So we have the gene, and then we have also the gene copy. To start with, they're the same, but over time, over lots of hundreds and thousands of generations, these two copies will become different. Maybe this one, so the color here represents the similarity to the original sequence. Maybe this one doesn't change very much. It basically stays the same. Maybe it retains the same function, but because we've got an extra copy, the other one is potentially more free to evolve, to mutate more freely, is less constrained, and it accumulates more changes over time. So this was kind of the classic view of protein evolution, that most of it happens through duplication, copying, and then divergence, changing over time. This is not just a theory, but there's lots of examples. One example that's been shown in SARS-CoV-2 is two of the genes in the genome, ORF7A and ORF8. They look quite different, but we can actually, through comparison with other genomes, we can actually basically infer their history. We can trace their history backwards and show that they originally, this one, ORF8, originally was descended from ORF7A. So the ancestor of this gene was copied, and then it turned into these two different proteins in some coronavirus genomes, ORF7A and ORF8. And there's lots of examples of this duplication divergence. That's the standard mechanism by which proteins evolve. So we can also visualize it in a kind of standard evolutionary diagram of a tree. So we imagine we have this ancestral protein, and then it changed over time. Maybe this tree represents like 1 million years of evolution. And these are all the different descendants of this ancestor. So different colors because they've changed in different ways over time. These are the modern proteins that exist today. So this would be kind of standard protein evolution, how you could visualize it. You could also imagine that maybe this protein got copied somewhere in the tree like here, and then all the descendants that come from this part of the tree would have this copy as well. So this was the standard duplication divergence picture. This is what we used to think. Sometimes it's been described as the Big Bang model. So that's the idea that nearly all the proteins in life, whether they're in E. coli, bacteria, or humans, or mice, or plants, nearly all the proteins we find can be traced, have an ancestor back near the origin of life. So that protein evolution is just duplication and divergence happening over millions of years. There were good reasons to think this, and this was the standard view. And there were good reasons because it is true for a lot of the proteins, like I just showed with ORF7A and ORF8. An example of this is these two protein complexes that look incredibly similar. This is ATP synthase, another key molecular machine in cells across found in all of life. They look basically the same, but actually this one is from a cow at the top, and this one is from bacteria E. coli. So the moral of this story is that some proteins don't change very much. There are slight changes. There might be one or two proteins different in this protein complex that have arisen through duplication and divergence, but basically they've stayed the same. And this is true for some proteins, but it's not actually true for all proteins. So as we got more data, it turned out the picture of protein evolution changed and became more complicated. So a lot of this material that I'll present from now is quite new, and it's still being explored. And it's useful to realize that science is constantly in development. We're constantly learning new things. And some of the most interesting areas in science are actually those which are still being worked out. So we're starting to understand some things about the new picture of protein origins, but there's still a lot of open questions, which means there's still a lot of interesting science to do. What changed? What is this data that changed? Basically, it's genomics, studying the DNA of different organisms. And this has really only been possible at huge scale in the last 20 years. So the Human Genome Project, and a third of this was completed at the Wellcome Sanger Institute. This massive project was completed in about 2003, and with the first human genomes. And this opened the doors to being able to sequence lots of other organisms as well. Before that, we just didn't have the technology to do this at high speed and high scale. Now, almost 20 years later, things have changed hugely. We have thousands of genomes. There's a new project which has started called the Darwin Tree of Life Project. And this project aims to extend this to sequence all the animals and plants in the United Kingdom. So this is a hugely bigger scale that people probably couldn't have even imagined when they did the Human Genome Project. But now we can do it because the technology is available. So what changed is that we have thousands more genomes that we didn't just 20 years ago. Now that we have these genomes, we discovered that a lot of genes actually don't fit this picture that we thought with the Big Bang model of protein evolution. This is illustrated nicely here. This is LUCA, the last universal common ancestor. And we imagine that it had a few genes, so some of them are pictured here. Maybe it had like a few hundred genes as compared to the few thousand in most organisms today. So we can compare the genes that we find in LUCA to the genes we find in modern day organisms, shown here, lots of different colors that aren't found in LUCA. What we can infer from this is that a lot of these genes don't actually have an ancestor in LUCA, that they've arisen more recently. So many genes are only found in a small taxonomic group. So imagine that this part of the tree is just one genus or one family, or maybe just one species. So it's a small part. And this is a nice paper which gives the evidence for this, the fact that most genes are actually quite young and not too many actually fit the standard picture of the Big Bang model. So then this raises the question, okay, how did we get these new genes? How can a gene be born? There's the standard method that I've already talked about. You have the ancestral gene, and then it just diverges over time, maybe with a copy along the way. Sometimes this can explain the pattern that we saw before, that maybe you just can't, they do actually share an ancestor, but they've diverged so much you can't see the similarity. But as we dig into the details of this, we see that this only explains some of the data. And for other genes, we actually need to understand how you can get a protein coding gene from a sequence which didn't used to encode a protein. So the ancestor was non-genic, means it's non-coding. This is called de novo emergence. De novo means from nothing. So proteins that begin to exist, that didn't used to be proteins, instead they used to be non-coding sequences. This raises some questions which we can now think about. So to start with, we have a poll. Approximately what percentage of human DNA do you think is used to make proteins?
So you should get a little poll question jumping up on the screen that you can vote on. If you're on mobile or tablet and it doesn't show up, you can just type an answer in the chat box instead. Okay, we're getting quite a mixed response. There's at least one answer for every option. Currently, the most popular two are 2% or 82%,
Okay.
but it's really very split. 2% is now winning, but yeah, it's a very, very yeah, there's answers for all of them. It's fairly well split.
Yeah, so the correct answer to this is only 2%, and maybe some of you Googled to find that answer. But it's a really small amount. So there's a lot of DNA in the human genome which is not used to make proteins. So then the next question is, which you can type an answer to in the chat. What do we call the DNA that doesn't make proteins? The other 98 or a bit more % of the human genome. And then secondly, question B, what else does DNA do apart from encoding proteins? What's the stuff aside from making proteins that this 98% plus actually does?
Awesome, we've got quite a good selection of answers. So for part A, we've got some people saying introns, which is a really, I completely see where we're coming from that. I don't think it's the answer you were aiming for, Zachary, but I think it's an interesting point to cover in a second. Some people know it, so they've said non-coding DNA. Some people have put the dreaded J word, which I'll let you explain in a second. But yeah, a mixture, I can see Fran's face, a mixture of introns, the J word, and non-coding DNA. And then in terms of what else it does, lots of ideas around regulating things. What else have we got? Someone's put parasitic genes. That's not a term I know particularly well, but you're nodding, so I'll let you explain that in a second. Someone's put allows for easy evolution for part B, which is interesting. Regulates transcription, regulates gene expression. So yes, I think on the whole, regulation and some other jobs by the looks of things. So I will let you explain the J word.
Yeah, those are all great answers. And just on the parasitic DNA, sometimes it's called selfish DNA or various related terms. That is a subset. So there's a lot of the human genome that originally came from like viral sequences, and it just seems to be replicating and not actually to be functional. It's just replicating kind of for its own sake, without actually contributing to the function of the genome. Yeah, so yeah, lots of really good answers there. Non-coding DNA. Some regions of non-coding DNA have sometimes been called junk DNA, which is a really kind of controversial term. Now it's known that there are many different things that non-coding DNA can do. So it was called, some people have called parts of it junk DNA because it wasn't doing anything. So it was just rubbish. It was just evolutionary junk, and it didn't have a purpose. Okay, we'll put that aside for a second, come back to that in a little bit. What other stuff can the non-coding DNA do? It can do lots of different things. And like lots of people are saying, there's different regulatory functions. So they regulate the expression of protein coding genes, all different kinds of RNAs, particularly in the human genome or in other mammalian genomes. There's lots of these different kinds of RNAs. And I think there's a lot more that I didn't list that could be listed. And to be honest, I don't actually know what some of these do because I don't study human genomes, or animals. Bacteria are fortunately a bit more simple, but there's also lots of RNAs in bacteria that we don't really understand what they're doing. Okay, so what about this junk DNA word? We don't actually know what percentage of the human genome is functional. So something like 2% encodes proteins, another few percent encodes some of these different RNAs. There's also introns that people mentioned, but that only gets us up to a certain percentage of the genome, depending on exactly which criteria you're using. Like, is it transcribed? That's a certain percentage, or does it bind to proteins? That's a certain percentage. Just because it's transcribed or binds to proteins, it doesn't guarantee it's actually functional. And so it's a massive debate, actually, how much of the human genome is functional, and it will depend what we mean by function. This is somewhere that philosophers of biology have something to say about, and people can look into this if they like. There's even a nice Wikipedia page on non-coding DNA that mentions and discusses some of these things. So as far as I can tell, it's definitely still debated whether junk DNA is a useful term at all. It's a complex debate, but the main thing to take away is that it can't be used to describe all the non-coding DNA. So because we know that a lot of the stuff that's non-coding is doing these different things. How much of it is actually useful in one of these senses or another sense is still an open debate, which is interesting, and it kind of goes back to that point I said before, that a lot of science is still being worked out, and we don't have all the answers to. Okay. How is this non-coding DNA relevant to the new genes topic? Well, this ties in with the question, how can a new gene be born? How can you get a new gene? This is one of the best examples that has been studied. This is illustrating an antifreeze protein, which is found in some fish. So fish which live in Arctic or Antarctic waters often need proteins to stop their blood freezing, basically, when it gets really cold. They need some kind of way to survive when it's extremely cold below freezing levels. And some fish have evolved different kinds of antifreeze proteins. And some of these have been studied and been shown that actually because they're only found in these fish, like they're not, if you're an elephant, you don't need an antifreeze protein. You know, if you're in lots of other places that isn't Arctic or Antarctic waters, you don't need an antifreeze protein. So these proteins are taxonomically restricted. They're only found in certain fish that use them. And that raises the question, how do they get there? Did they adapt from a common ancestor protein? Turns out often they didn't. They actually adapted from non-coding sequence. And this is what's illustrated here, that these fish in this part of the tree just have non-coding sequence, or maybe they have maybe they have a small protein or a part of a protein. But the ancestors, if you trace back the phylogeny, if you work out what the common ancestor must have been, it must have been non-coding. And then over time, they kind of accumulated protein elements until some of these fish have this kind of full antifreeze protein with all the elements of a gene. So what goes up to make a gene is quite complicated. It's not just the protein, it's also some of these like regulatory elements. And over time, because of selective pressure huge selective pressure for this antifreeze protein, because without it, the fish couldn't survive in these really cold waters, these proteins originated and got selected. So yeah, this is one of the really nice examples. Because it's been studied in a lot of detail, we can show really clearly what the ancestor sequence of these genes looked like, and it turns out it was non-coding. So in order to understand the transition from non-coding DNA to coding DNA, it's useful to realize that not all non-coding DNA is the same. So it's not just like any random piece of DNA is likely to become a new gene, but some bits of DNA are more likely than others to become a new gene. So this is quite technical, but basically, if you want to understand how new genes come about, you need to kind of go into some of these details. So an open reading frame, you may have heard this term, an ORF, is any part of the DNA between a start codon and a stop codon. There's lots of these that aren't transcribed or translated. So just because a bit of DNA has a start codon and then downstream it has a stop codon, that doesn't mean it's going to turn into a protein. So there's lots of these that aren't transcribed, they're not turned into RNA, they're not translated, they're not turned into protein. But they're still there, they're still an ORF, so they still could potentially be a protein if they got transcribed and translated. And this is basically what's behind the evolution of new genes, is that there's lots of these ORFs lying around, and some of them turn out to be more useful than others. more likely to be translated or more likely to fold into a functional protein. Different open reading frames have different properties. They have different codon usages, so they have different codons in them. They have different GC content, so they have different nucleotide patterns, different likelihood of forming a folded protein if they are translated, and these kinds of things. So this is mostly what I study in bacteria. What kinds of sequences are more likely to be involved in creating new genes? Also, many open reading frames are overlapping other genes. So then it gets really complicated. They're not just in between genes, but because the DNA letters are read in triplets, because codons are made up of 3, that means that you can shift the reading frame and read a different triplet. And that allows you to have sequences which use the same DNA, but they have different, they read different triplets, and that means they encode different amino acids. They encode each overlapping each other. These overlapping sequences are generally ignored, but they have some really interesting properties, and this has been the focus of my research. Some of these properties are given by the structure of the standard genetic code. And with these 2 pictures, I just wanted to illustrate that you may have seen a picture like this for the code, but usually you wouldn't see the colors. And the colors represent the chemical properties of the amino acids, which are encoded by the code. Now, it turns out that these are clustered together. So all of the codons in this column have this particular fit into this chemical group, the amino acids. These ones are charged and they fit into these groups. So the way that the code works, the mapping between nucleotides and amino acids means that the amino acids are grouped together according to their chemical properties. One of these chemical properties is hydrophobicity, how much the amino acids are likely to dissolve in water, basically, or their kind of affinity to water. So these ones are hydrophobic and these ones are hydrophilic. And this is just, again, illustrating that there's these patterns in the structure of the code, and this is going to have impacts when we think about what happens when you have overlapping reading frames. So that was quite technical, but the main point is that different non-coding sequences have different chemical properties, and part of this will be determined by whether they're overlapping other sequences that already exist.
And just a quick question from that last slide. Someone's asked a question that I think is really great, saying,
okay,
why are the ORFs there in the first place? I don't know if there's an answer to that, but I thought it was a great question.
Yeah, so try and go back. I'm just trying to go back on my slides.
You press your left key, it might.
Oh, yeah, okay, that was, yeah, okay. Why are these all together? Yeah, so some of this is just random, that if you have a certain proportion of different nucleotides in a sequence, then by chance you'll occasionally get an ATG appearing, and by chance you'll occasionally get a TAG or another stop codon. So an ORF is just this region between an ATG and a stop codon. And so because you only have 4 letters that you can use, A, T, C, G, with a long enough genome, you end up getting a lot of these start codons by chance and a lot of stop codons by chance, many more than you actually use to make genes. So like in the E. coli bacterium, it has something like 4,500 genes, but it has like 4 million letters. So there's a lot of possibilities to have these short open reading frames. So yeah, the short answer is just by chance, basically. There's lots of open reading frames. And then if these did get translated, they could turn out to be useful. And this is what evolution kind of uses. Okay, so not all non-coding DNA is the same. Another example of this is just a really simple illustration of the genes in part of a genome. So imagine this is like 10,000 nucleotides or something. Then you have a bunch of genes on the plus strand here and also on the minus strand here. These are the genes that are annotated, that we know they do something, but actually these little ones here, these are illustrating the open reading frames. And there's lots of these short open reading frames that aren't actually annotated as genes. And that's because just by chance, there's a start codon here and a stop codon here. So there's thousands of these that aren't genes, but they could potentially be genes if they were translated. And the main point of this is that these open reading frames actually have different properties. You can score them in different ways. They might have different, they might encode different amino acids, all that kind of thing. This is just a particular example, a predictor, which is used to work out which, because it's quite a tricky problem, which of these open reading frames is actually a true gene. And we can score them in different ways. And this is how we've decided these ones are the true genes. Mostly, if you look at it carefully, you see these are the longest ones. But actually, there's a lot of these short ones that could also be true genes. And these green ones, along the x-axis is a score that's been given. The green ones are the ones that have been annotated as genes. We say these ones are the genes. And usually they have the highest score. So that makes sense. But then these are all the ones that weren't annotated as genes. But still a lot of them have quite a high score. So in other words, a lot of these things that aren't officially genes, they still look like genes in some way. This is what I've called the shadow zone, that there's kind of it's not just a dichotomy, either you're a gene or not a gene, but there's a lot of sequences in the middle, these non-coding sequences that I've talked about in the shadow zone. And this is what evolution can then work with and create new genes from. Okay. That was the theory. So a couple of practical examples from SARS-CoV-2, the virus which causes COVID-19. We've mentioned one of these before. This was the genes ORF7A and ORF8, which are pictured here again. These two turn out to be related to each other. That ORF8 was actually ultimately derived from a copy of ORF7. And the same happened with these two genes, ORF3A and M. This gene ORF3A was a copy of this gene, the M gene, the membrane protein. And so this is a common thing which happened in evolution, and we can, for some of the proteins in SARS-CoV-2, we've worked out how they got here from ancestors, which we can trace back. And this is the kind of research which, some of the research which I'm involved in. I've particularly focused on the overlapping genes that I briefly mentioned before, both in bacteria and viruses. And this is a nice illustration of what I was talking about with overlapping genes. Because the DNA or RNA is read in 3 letters at a time, this means that these letters read like this, 1, 2, 3, 1, 2, 3, will encode this protein. In this case, this is the nucleocapsid protein, SARS-CoV-2, one of the most important proteins. But it turns out that actually embedded within this protein sequence, embedded within this gene, is this other sequence which actually encodes a completely different protein, ORF9b. This is really surprising. Not too many people have thought about these things, but this protein is also known to have a function and is actually a real protein. We know the structure and we have some idea of what it does. So there's a lot of these kind of hidden overlapping genes which are embedded within the non-overlapping genes. They're kind of usually older and better known genes like this nucleocapsid protein. And these are what I've studied, both in SARS-CoV-2 and also in bacteria. In this case, the 2 genes here. So there's a known gene called TLE3, which was on the minus strand. So in one of the 2 DNA strands, then on the other strand, the plus strand, again, embedded, we have this overlapping gene. And in this case, we showed that it's transcribed. RNA is translated with a method called riboseq. It's also translated with a method called mass spectrometry. And so we could show these really are real genes that are producing proteins from both strands. This is just another illustration of the kind of thing I'm thinking about, the sequence diversity. So the kind of changes that happen in the different reading frames. So there's 3 possible ways you can, because it's read in triplets, the genetic code, there's 3 possible ways you can read on the plus strand, and there's 3 possible ways you can read on the minus strand. And that's why you end up with 6 different reading frames, the 3 plus strand, the 3 minus strand. And then you can look at the way that each of these genes is evolving, basically. Okay, so now I'll summarize. Evolution often makes new genes. We have some clear examples of this, but a lot of them are not yet well understood. So there's a lot left to discover about what makes different organisms different from each other and the roles and mechanisms of new genes. Okay, and these are some last questions you might want to think about. Why are proteins important in the first place? How do you personally think about evolution? How do you visualize it? Maybe now you have a better idea of what's the link between evolution and proteins. Why does the evolution of proteins matter anyway for understanding them? And are there maybe some things about evolution that we can never understand as we keep on learning more and more? Will we eventually find it all out, or is some stuff always going to be a bit mysterious? So on that, maybe we go to questions.
Yeah, we've got loads of really good ones. So we'll take some questions and then we'll let Zachary talk about his career. And then if there's any career related questions, we'll do those afterwards. So I think my sort of favorite one that came in quite early, linking to why you specifically use like bacteria to study this. Someone's asked, are bacterial mutations more noticeable because they're a smaller organism with fewer genes? So less mutations are needed for a more obvious change.
Yeah, that's a really good question. I actually don't know the answer to that. So I don't really have a good answer to that, but it's possible. But at least what is definitely true is that we can, because they're so small and because we can have like 1 billion of them in a test tube, we can do all kinds of interesting tests with them in ways that we couldn't test other cells, or partly for ethical reasons, but also partly just the practicalities of large scale and short time frame mean that we can study a lot of things in bacteria that we can't study in the other things. So we can actually work out at the molecular level what the mutation is doing in a way that is harder to do with other organisms. But yeah, I don't know whether we actually know the answer of whether the smaller genome, I mean, I guess smaller genome means fewer genes. So probably each gene has a bigger impact. That's a sensible, I think it's a sensible idea, but maybe someone needs to test that.
There you go. There's a whole science project you can go off and do in the future if you want the asker of that question. Another really interesting question asked, would a protein go extinct if mutations happened at a rapid scale? So I guess if one gene mutated a lot, would that protein just stop being a protein?
Yeah, so there's a related idea called mutational meltdown, which is important for viruses. And this is actually this is of interest recently, because this is the mechanism by which one of the anti-COVID drugs works, called molnupiravir, I think. And it works by mutational meltdown. So it works by creating lots of extra mutations, by increasing the mutation rate. And that because the mutation rate is so high, the virus basically can't survive. It accumulates too many mutations. So yes, at least for an organism, if the organism has too many mutations, then it's not going to be able to continue to evolve and survive, and it would die out. And in viruses, this has been studied a bit, and it's called mutational meltdown.
So we've had loads more come in, so I'm sort of trying to scroll through and grab out some really good ones. So someone said, you mentioned earlier about more technologies being available to study genomes. So what type of technologies are you referring to? And I wonder, are there any specific ones that you use in your research that are kind of newish and have like helped sort of bacterial studying get easier?
Yeah, so the main thing I was thinking of with new technologies is just that we can sequence the genomes in huge scale. So genome sequencing, like to sequence the human genome, initially it cost like $1 billion US, and now a human genome costs something like $1,000 US or less. And so that's partly just the scale has increased. There's a lot more of these machines available, and because they're being produced at scale, they're cheaper. So that's the main thing, but there's also lots of associated technologies. So one of them that I use is illustrated here, this thing called RiboSeq, ribosome profiling. So this is a subset of sequencing, which looks at the RNA, which is covered by ribosomes, and looks at the RNA, which has actually been translated. But there's a lot of these different like RNA sequencing technologies, different kinds of DNA sequencing technologies. Yeah, so it's a massive field, and there's a lot of different technologies that are still being developed, which is reducing the cost, increasing the speed, increasing the number of organisms we can look at, that kind of thing.
Awesome. I just popped in the chat box an article that we have that basically compares the major types of genome sequencing over the last 50 years. And at the bottom, there's a really handy table that just shows how much quicker and better it's got, so that, yeah, studies that would have taken years can now take like days or weeks rather than, yeah, years. So that's obviously made a huge difference to projects like yours. Right, I'm trying to pull out some other good ones. Someone asked about the shadow zone, which I think sounds nice and mysterious. Is there any sort of ideas or explanation about what their role might be, sort of more widely? If they're sort of, do they have like do we think they might have a role even if it's not becoming a protein?
Yeah, so not everything necessarily has a role. Some things are just the way that they are, and evolution has maybe made use of that in some way, or maybe not. So whether the shadow zone like has some kind of role is an open question. And so this stuff is all of the kind of second half of the presentation is stuff that's very much still in development and might be kind of controversial, or like less clear. So this whole idea of the shadow zone, what we mean by this, how useful it is, this is still kind of being developed. And yeah, the answer to most of the questions about this kind of stuff would be we don't know. But it's interesting to try and find out.
And someone's asked the classic chicken and the egg question. So if genes code proteins and proteins help to synthesize genes, do genes appear first or did proteins appear first?
Another good question, which the best answer we have is we don't know. And because there's different, so this goes back really to the origin of life, and there's different models of that. And some people would say maybe genes were first, like RNA world hypothesis, and others will think that maybe there was like a peptide RNA world where so there was kind of a bit of both. And the best answer we have is we don't really know, and there's some models, and people are working on trying to make sense of that. Yeah, at least I don't know.
No, I think that is the reigning, yeah, it's one of those, we don't have a time machine, so probably never know for sure, but yeah, it's all about sort of, yeah, modeling and better predictions. Someone asked, as you were going through the slides, about convergent evolution as compared to divergent evolution. I wonder if you just wanted to touch on that briefly.
Yes, so that's a massive issue, so probably useful to see a tree somewhere. So convergent evolution would be where a similar form, like a similar phenotype, maybe a similar protein ends up evolving from two different ancestors. And so convergent evolution usually happens at the larger scale, like in terms of the whole shape of the organism. Like dolphins and sharks, for instance, look quite similar in some ways. They have a similar overall shape, even though their recent ancestors are quite different. Convergence can also happen at a molecular level, but it's one of the things that's kind of being studied quite a lot, and we don't have a good understanding of. Usually evolution, because mutation is in some sense random, or it's not precise in the effects that it has, if you run an evolutionary experiment over time, like we see way back here actually, the different lineages would diverge, would be the tendency. Occasionally they may also converge on the same kind of outcome. Like it might be that this lineage, which went here, maybe it also maybe ends up being the same mutation as this lineage here, because that mutation is useful for surviving in antibiotics. That would be an example of convergence. But most of the changes, if we look for it, will be divergent. And yeah, so evolution will be a mix of both.
Awesome. I think looking at the time, I might ask you to share a little on your career, because we've had a few questions about what did you do at uni, which I know you've got in your slides. So if you've got questions as Zachary's going through about why he picked science, I'm sure he's going to touch on that, but pop those in the chat box or Q&A, and we'll pull out some in a second as well.
Yeah, so what's useful for different people will be very different. I can just give a very quick overview of what I did, and then everyone else in science will have sometimes a completely different picture of what they did. So don't take what I did particularly seriously, other than to see the kind of diversity that is possible. So in my case, I'm from New Zealand. I studied what's called a conjoint degree, which is, I think, more common in New Zealand. So I studied both arts and sciences. I was interested in philosophy, so I studied philosophy, economics, and biology. And then I decided for various reasons to focus on the biology, partly because the other ones, or at least philosophy, wasn't going to give me a job realistically, and biology was, and I'm really glad I made that decision. So I focused particularly on molecular biology in a postgrad diploma. And then I started a master's project that's turned into a PhD, actually in experimental evolution, which is similar to this animation that I showed years ago. This one is an example of experimental evolution. In my case, I was working with yeast and data rather than bacteria like in this one, but similar, not this mega plate, but also looking at a few hundred generations to see what changes happen. So that was my PhD in New Zealand. Yeah, then I moved to Germany, a couple of different places. Most of the time I was in Germany for a few years, was in Munich. So after I finished my PhD, I immediately took on what's called a postdoc, which just means after PhD research. And I was doing research on bacterial evolution. I was briefly at another university in Germany, then I came to the Sanger Institute. If people wonder what's the long-term kind of trajectory of this kind of thing, I would like to lead a group. And so that means I would have PhD students and postdocs that work with me on some of these topics. These are great experiences, but I wouldn't want to say this is like for everyone. I wouldn't say it's like easy and purely fun. It's kind of it's a mix. And most people are honest about science would say that as well, that there's lots of advantages, like interesting research, the ability to travel and explore new things, but there's also disadvantages in my case that I had to go to the other side of the world to do it, in a different culture and that kind of thing. So there's always pros and cons. So this was Munich and this was Karlsruhe. This was an example of what happened when someone got a PhD in Munich. They kind of celebrated in this way. This was a group that I was part of for a few years. This was the campus that I was at the Technical University of Munich. Lived in a really nice town called Freising, which is a medieval kind of village just out of Munich, which looks like this in winter. So it was a really nice place to live. But now I'm at the Sanger Institute, and this is the group that I'm part of here. And this is I mean, Munich was great, but the Sanger Institute, I would say, is an even better place to do science with a lot more opportunities. So I'm really glad to be there. I was asked to maybe give some tips, so came up with some things. The main thing is every path is different. Science is very broad, so it's very hard to give advice. But these are some things I think are general, at least for the kind of science that I do. Firstly, being willing to learn new methods is always helpful. So maybe, actually an example of this which just comes to mind is a PhD student who I supervised who just finished. He started as a physicist, and then he ended up doing some of the stuff I was talking about the genetic code and evolution of new genes. So he transitioned from physics to biology. So, and this happens actually quite a lot, that people do change fields. So don't be too worried about exactly what you should study to end up where you want to go. People do actually change. And the main thing is if you're willing to learn, and if you have some basic skills that are useful across science, like coding is really useful, statistics is incredibly useful, and just being curious and reading lots of things and being interested in a topic, I would say is often more important than what specific skills you have, because you can pick up new skills once you've got the basics of some kind of science degree. And just always being willing to ask people for help is the main thing, because basically science is always changing. You need to be flexible. You need to be willing to learn new things. So those are my quick tips for a career in science.
And thank you. We've had a really lovely question that I think might be nice to get your sort of input on. So someone's asked sort of how many people are involved in the research team in different areas and what type of skills. So I presume you work sort of as what we'd call a wet lab scientist, but I wonder if you could tell us the sort of people that you interact with that have quite different jobs to you. So yeah, who do you come across in your in your work?
Yeah, so I actually don't do any wet lab stuff. I did a little bit during my PhD, but basically everything that I do personally is computational. I'll go back to this just for an example. So this was a really large project to get this kind of a diagram and some other things that were published in this paper. So a lot of this was wet lab work. So the main PhD student who was leading this, she was doing these sequencing technologies like RiboSeq, RNA-seq. I was doing other evolutionary analyses that went on top of this, and some of the bioinformatics behind this as well. So yeah, there was a mix of like computational people like myself. Also, I particularly had an evolutionary focus, but other computational people would have a different focus, like maybe the sequencing focus. There were people who were experts in this small part of the diagram, which was mass spectrometry. So they were focused on finding peptides in samples of cells. And they didn't really know anything about this area, the ribosome profiling. And the people who did the ribosome profiling didn't really know anything about the mass spectrometry area. So there's lots of really specialized people who are contributing to the overall project. And I didn't have much to do with these things, but I was doing the evolutionary analysis. So yeah, it was a real mix of different wet lab skills and analysis and other computational things. In other projects, like I said, the PhD student had a physics background, and that turned out to be useful for some of the theoretical modeling and coding stuff we were doing. Yeah.
Awesome. And then there's also, always a side that there's got to be people that run the lab and sort of, make sure you've got your equipment in so that people can actually do their experiments. So there's lots of different, I guess, what we might think of as sort of supporting roles that aren't maybe doing the core science, but are making sure that, the lab stays open, you've got your kit and, everything's in the right place and legalities are kept to and all the rest of it. So there's lots of sort of wider jobs somewhere where we work that keep all the science happening so that people like you and people in the wet labs can actually do the science they're doing. So yeah, there's if you're worried that one bit of science might not be for you, there's other sort of ways you can be involved as well. I think those are the only sort of career specific questions. So if anyone else has a career-y type question, pop them in the Q&A. If not, I'm going to pick out a last few sort of sciencey ones because we had so many good ones. I'm going to have to pull out some more. Someone was particularly surprised that viruses, you were saying about the overlapping genes. Someone said, in viruses that makes sense because, they have to fit all of their genome into a capsid. But why would bacteria have overlapping genes? I think this was, that they were particularly surprised by this bit of information.
Yeah, this is a really good question. This is actually the kind of response that a lot of people who are reviewing our papers would also say the same thing. So this is a standard view that a lot of people have, is that viruses have overlapping genes because they need to pack a lot of information in. And there's a strong selection pressure for minimizing the genome size, and other things don't have overlapping genes because there's no pressure for that. This was the standard view. Actually, there's been a major study and various other related things that have shown that viruses don't really have the strong pressure for minimizing the genome size. So there is quite a flexibility for viruses. And also that there's lots of overlapping genes. So there's overlapping genes in humans, and there's overlapping genes in lots of other organisms. Maybe particularly bacteria, but definitely bacteria and these other things. So then the question is like, oh, why do overlapping genes exist if it's not just to pack in the information? And probably the main answer I would say is because it's to do with this stuff that I work on, the evolution of new genes, is that it's a good way to get novelty. And there's various reasons for that. People can read our papers if they're interested. But yeah, overlapping genes seem to be involved in the origin of new genes, exploring evolutionary novelty, rather than just packing the information in.
So that's really useful. One about sort of future technology developments that you may or may not know about, but I'll ask anyway. What are some technology developments that could help from a genomics point of view? For example, quantum computers. I don't know much about this. I don't know if you do.
No, no, I don't. But any computational advance will be useful for genomics because we're always kind of pushing the limits of, I mean, it takes a long time to run some of the analyses. So things like some of the protein structures that I showed, I think, I'm not sure, at least potentially, they have actually been calculated, not from like crystallography, but actually they've been computed. They're actually predictions rather than actual visualizations of proteins. So this is called AlphaFold. It's the most famous example. It's a technology that Google basically developed to predict protein structures. And these kinds of things require computational advances. So there's definitely huge room for people with different kinds of computer science expertise to work on biology. And yeah, massive opportunities there. Maybe quantum computing will have some role there, I have no idea. But yeah, there's heaps. If you can code and you're interested in biology, that's a really useful combination. And I'm pretty sure you'll get a job somewhere. So I encourage that.
So I'm going to pop a link to the AlphaFold in the chat because I think it's, oh no, that's the wrong people. There we go. I think it's absolutely amazing. Essentially, it's an AI that can predict how proteins fold, which is not something that people can do or computers could do before. So it's I think it was seen as the biggest challenge in biology in the last sort of 50 years. So really interesting. So if you're interested in that sort of intersection between cool new tech and biology, definitely worth a read. Just gonna scroll through some last couple of questions. So someone's asked, how exactly do you see these proteins and know that they've changed? Because obviously they're too small to just look at. So I presume you use lots of different methods to visualize them, but could you sort of tell us about a couple?
Yeah, so there's a couple of different ways you can think about that. Firstly, you can just see that you can look at the DNA sequence and see what mutations happened, and then you can predict, okay, this DNA mutation would cause this amino acid change. So you can see what's happening to the sequence. And this is the main thing that evolutionary biologists have traditionally looked at. But now, because of AlphaFold, but also developments which have led to things like the bacterial flagellum that I showed, these structures as well, I think. Some recent developments like called cryon electron microscopy have allowed improvements in visualizing the protein structures as well. So yeah, there's a lot of work on the sequencing side, but there's also a lot of work on the structure side to get this kind of impressive image. So we can see the changes at the sequence level, but we can also start to predict or actually even visualize the changes at the structure level.
Awesome. I think we've got through most of the questions. I'm going to pick up the first one that came in. So apologies to this person for waiting so long. But the very first question that came in was when you explaining what a genome is, and they've asked, does that also include mitochondrial DNA in the cell? So I guess that's for sort of animal ones rather than.
Yeah, so I think sometimes these are called the mitochondrial genome. So in some sense, that's a genome as well, but it kind of depends on your definition. But yeah, so sometimes eukaryotic cells have these organelles. They have their own separate bit of DNA, which is either chloroplast or mitochondrial. And yeah, they also have kind of a genome of sorts, but it's not, in some ways, it's not a genome in the same sense because it's not encoding the whole information for the cell. It's just encoding a few proteins which are used by the mitochondria.
Awesome. I think that is pretty much all the questions we've had. So we're going to, I think, wrap up there. Zachary, thank you so much for joining us. I've definitely learned lots of new words and loads of cool concepts as well. So thank you so much for joining us. Let me just share my screen to finish off then. So as I said, this has all been recorded and will be available on YouTube and on our website probably tomorrow, if not Monday, and in your follow-up email from Eventbrite, you'll get the link to that. So if you've missed any of the start, don't worry. Our next session will be at the end of March, and we'll be exploring the sequencing of the malaria parasite. So that's going to be a really interesting talk from a researcher that's looking at a specific part of the malaria genome that isn't well understood. So the link and information for that as well will come out in your follow-up Eventbrite email. So thank you very much for joining us, everyone, and hopefully we will see you next time.